Good day. My name is Janine Post, and I work at the Developmental Bioengineering Group at the University of Twente. Today I would like to show how we use a combination of computational modeling and molecular cell biology to investigate the molecular mechanisms of cartilage development and disease. Our research is focused on the mechanism of osteoarthritis. 1 out of every 10 people in the Western world suffer from osteoarthritis. That is over 150 million people worldwide. The symptoms are pain, loss of function, stiff joints and swelling. And although there are risk factors described, like age, obesity and trauma, the real onset of osteoarthritis is poorly described. There is currently no cure. However, there is some treatments which exist of pain relief and total joint replacements, of which about 300 million per year happen worldwide. We do know that osteoarthritis, or OA, is a gradual process, and it is characterized by loss of structure and loss of cartilage-specific proteins, such as glycosaminoglycans and collagen II. At the same time, we see an increase in hypertrophic markers such as collagen X. So these are small pieces of cartilage sliced, and histological stainings for glycosaminoglycans are performed, and immunohistochemistry for collagen II and collagen X. And what you see is that this is grade 0, or healthy cartilage, and you see a smooth lining and a nice even staining of the glycosaminoglycans. And you see as osteoarthritis progresses, you see a quite rapid loss of structure and staining, and an increase in collagen X. We already know a lot about the different cells, signals and transcription factors that are involved in cartilage formation. During development, cartilage is formed from the mesenchyme and under strict regulation of the transcription factor SOX9. Permanent cartilage is formed that lines the long bones in articular joint. In contrast, in the growth plate, cells are under regulation of the transcription factor RUNX2 and undergo hypertrophy. This is followed by cell death and bone replacement. Similar processes of hypertrophy and bone replacement are found in OA. There is some information about the signals during differentiation and hypertrophy, but nothing is known about the onset of OA. There are no human tissue samples available to study this, or indeed not enough. And we have previously found that the Wnt and BMP antagonists, FRZB and GREM1, are highly expressed in articular cartilage, but not in growth plate cartilage, and that they do play a role in preventing chondrocyte hypertrophy. And in this work we want to investigate whether these factors play a role in preventing OA. We all know that not expression, but protein activity is important for determining the cell function and cell fate. I want to illustrate this for cartilage. We have measured the gene expression of SOX9 and RUNX2 during OA development. You see here SOX9 and here RUNX2. As you can see, the expression of SOX9 stays high during the first stages of OA, then drops rapidly at later stages. As expected, RUNX2 is not present in the early stages, and its expression increases gradually with OA progression. However, this is gene expression. If we look at the protein activity of SOX9, which we can do by looking at the expression of its target gene, aggregan and collagen II, we observe a gradual loss of gene expression of these matrix proteins, but much earlier than the decrease in SOX9 expression. This indicates that although SOX9 is still highly expressed, its function or its activity is decreased. As expected, genes correlated to the activity of RUNX2 are now seen much later during OA. So this shows that not expression of SOX9 and RUNX2, but the protein activity determines the outcome. And it is therefore important to investigate protein activity. Ultimately, protein activity is a result of a dynamic signaling network. Previous research by others and by the DBE group has unraveled many important mechanisms of articular cartilage homeostasis. Many of these mechanisms involve signaling crosstalk. The current issues are cells receive many signals at once and integrate this into a cellular response. We have large amounts of data, and we also do not have all the information needed to fully understand what's going on in cartilage. Ultimately, the signals are integrated in this black box that regulate the exact activity of SOX9 and RUNX2, or RUNX2. We therefore use computational models with experiments to paint a complete picture. For this we built ECHO, which stands for Executable Chondrocytes, a dynamic protein activity network. ECHO consists of 7 inputs, which are growth factors and cytokines. A total of 93 nodes, proteins and genes, that are connected through 290 interactions. And we know 2 attractor or steady states. SOX9, associated with healthy articular cartilage, and RUNX2, associated with osteoarthritis. In ECHO, we can simulate a switch from a healthy SOX9 positive state to a RUNX2 positive state and look at the changes of activity of all nodes in time. In the video you see the shift from a SOX9 positive state to a RUNX2 positive state. Addition of Wnt and BMP antagonists DIFFERENTIATION and FRZB and GREM1 that are known to be expressed in articular cartilage to ECHO resulted in stabilization of the SOX9 positive state. In this simulation, and in the corresponding heat map, we see that there are changes in the activity of a number of nodes, but no change occurs in the SOX9 positive state. In ECHO, we can also mimic stem cell differentiation by translating gene expression profiles of MSCs to starting values in the network and then simulating addition and removal of growth factors and cytokines. Okay, let's look at early onset of osteoarthritis and see what inflammation if inflammation plays a role in that. We already know that an increase in Wnt signaling is associated with OA, and we also know that inflammation is present. However, we do not know what the role of inflammation is in the onset of OA. After mechanical stress or trauma, cartilage can be damaged. It is possible that in a response to this damage, inflammatory pathways, such as the NF-kappa B pathway, are activated. In turn, they will upregulate expression of pro-inflammatory signals, interleukins, TNFs. This will create a positive feedback loop by activating more pro-inflammatory pathways, and this may eventually result in chronic inflammation. Despite having a large functional overlap in the development of OA, the mechanisms of interleukin-1 beta and Wnt crosstalk have remained largely unknown. Using a combination of computational modeling and molecular biology, we revealed a mechanism by which interleukin-1 beta upregulates Wnt signaling by downregulating Wnt antagonists, DIFFERENTIATION and FRZB. And this happened via INOS, since blocking INOS restored DIFFERENTIATION and FRZB expression to control levels. And this is shown here. So these are control levels. If we add interleukin-1, DIFFERENTIATION is greatly reduced, but after inhibition of INOS, it is back to the control level. In the case of FRZB, it's even expressed higher after INOS blocking of INOS. And this is true both for mRNA expression and protein expression. We made a model of this and used this model to predict this already, as seen in this heat map. So this led to a new hypothesis that addition of interleukin-1 beta to chondrocytes will induce the RUNX2 state by downregulated DIFFERENTIATION and FRZB expression, and thus upregulating Wnt activity. We tested this hypothesis in echo by adding the interleukin-1 beta signaling pathway to the network. And to test this hypothesis, we turned to FRAP, or fluorescence recovery after photobleaching, as we previously showed that FRAP can be used to determine changes in transcription factor activity. For this, we transiently transfected human chondrocytes with either SOX9 GFP or RUNX2 GFP and stimulated the cells with various cytokines. In theory, proteins can be in any of three states. Unbound, in which the protein mobility would be high. A more likely state, when it is present in a protein complex and the mobility is lower. And in case of transcription factors, it can be bound to the DNA with very low mobility. And although we cannot say for sure that when a transcription factor is bound to DNA, it is transcriptionally active, we do know that it cannot be transcriptionally active if it's in any of these states. So we therefore always compare a steady state unstimulated cell FRAP rates with a state in which we stimulate the cells with some kind of factor, either an activating factor for SOX9 or an inhibitory factor. So to illustrate the types of changes in transcription factor mobility we can measure with FRAP, I show some examples of changes in SOX9 mobility in response to various extracellular factors. Here we see recovery curves, so fluorescence recovery curves of cells in healthy chondrocytes. And the addition of the anabolic factor BMP7 in red slightly and not significantly decreases the mobility of SOX9. However, if we add the BMP7 antagonists, GREM1, we significantly increase SOX9 mobility, indicating a loss of transcription factor activity. We did verify this by measuring target gene expression and chromatin immunoprecipitation to show that there was a loss of DNA binding. Addition of Wnt3a significantly increased SOX9 mobility, which corresponded with a loss of SOX9 target gene expression and DNA binding. This could be rescued by the addition of the Wnt antagonist Dickoff and FRZB. Please note that not all of the cells in the culture respond similarly, as can be seen by all these individual measurements. And also not all donors respond to the same extent. We are now investigating the role of epigenetic markers and genetic variants in this donor variation. In addition, we have seen differences in FRAP rates between healthy and OA chondrocytes. Back to our hypothesis that addition of interleukin-1 beta induces a RUNX2 positive state. FRAP data show that there is a slight increase in SOX9 mobility after interleukin-1 treatment, both in healthy-looking cells as well as in osteoarthritic cells. So the interleukin-1 is the red line. Please note that these healthy-looking cells are isolated from macroscopically healthy-looking cartilage from patients with osteoarthritis. This is sometimes also called preserved cartilage, so it's not to be confused with actual healthy cartilage. We noticed that after interleukin-1 beta treatment, there was a slight decrease in the residence time of SOX9 on DNA, and that in OA cells there was a slight increase in the residence time of RUNX2 on DNA in terms of recovery half-times, which indicated a slight loss of SOX9 activity and an increase in RUNX2 activity in these cells. However, when we look at downstream effects using qPCR, we observe that healthy-looking chondrocytes showed no significant loss of collagen II and aggregan, while in osteoarthritic cells there's a significant downregulation of these genes. However, we did observe a slight increase or significant increase in catabolic gene expression for MMP1, MMP3, and MMP13. So in all these pictures, without the control is not untreated cells, it's in white, and the gray bars are the interleukin-1 treated cells. So what we do see is that the response of osteoarthritic cells, or the expression of MMPs in osteoarthritic cells, is much higher than it is in healthy cells. So it seems that healthy cells may be partially protected against the effects of the interleukin-1 treatment. So what difference in between healthy and osteoarthritic cells could explain this? We have found before that the factors Dickoff and FRZB and GREM1 are found in articular, but not in growth plate cartilage. In addition, we have found that the Dickoff and FRZB expression gradually decreases as OA progresses. For GREM1, this is not really true. At first it seems to decrease, but then later increases again. We therefore activated Dickoff, FRZB and GREM1 in our echo model in the presence of interleukin. We ran simulations, and the steady state that is reached after the simulations showed that there is no switch to a RUNX2 positive state. It is still a SOX9 positive state. However, we do see activation of some downstream genes, such as the MMPs. So while addition of Dickoff, FRZB and GREM1 prevented the loss of collagen II and aggregate expression in both healthy looking and osteoarthritic chondrocytes, expression of MMPs was not prevented by the addition of Dickoff, FRZB and GREM1. In addition, we observed prevention of apoptosis in the presence of Dickoff, FRZB and GREM1, as shown by these Tunnel assays, in which we see apoptosis after interleukin-1 addition, and this is much reduced in the presence of interleukin-1, Dickoff, FRZB and GREM1. So these experiments correspond to the model predictions. So this talk has shown you how integration of computational models and wet lab molecular experiments provide insight into mechanisms that are important for tissue development, homeostasis and disease. In Animal, a modeling tool, we built ECHO, in which we show how signals are integrated in chondrocytes. And in ECHO, we are able to show the timing and intensity of these signals and to mimic the mechanisms of OA development. We use our computational model to generate and prioritize hypotheses that we can test in the wet lab. I want to end with thanking the current and previous members of the DBE group and our collaborators, in particular Stefano Schivo, who was a great help in building ECHO and running ECHO, the patients of OCON for donating their tissues, and our funding agents. Thank you for your attention.